appreciate your time. Uh, okay, I think I'll begin. So it's known that immune cells exist in the healthy myocardium and they account for approximately 5 to 10% of the total cells in the heart, and that they can respond to hemodynamic load in chronic disease conditions that are tightly linked with atrial fibrillation. But what happens in the setting of acute disease, and how can responses between the chambers differ? And what happens in the setting of acute stretch? Well, this is my project. I'm assessing the stretch-mediated immune responses to conditions linked with atrial fibrillation. So a bit of background, atrial fibrillation is the most common arrhythmia seen in clinical practice, and it's characterized by chaotic, uncoordinated beating of the atria. The conditions with the strongest links to atrial fibrillation are all conditions which confer elevated filling pressures and myocardial stretch, particularly in patients with valvular disease. These conditions, which ultimately lead to heart failure, are shown to have an important immune component to it, as originally inferenced by the works of Douglas Mann. He showed that there were elevated circulating pro-inflammatory cytokines, such as TNF and IL-6 in heart failure patients, and that these were linked to worse survival outcomes in a dose-dependent fashion. More so, patients with atrial fibrillation are shown to have increased immune cell content and macrophage content in their atria. Over time, the chronic elevations in filling pressures becomes a stimulus for maladaptive cardiac remodeling, including hypertrophy, fibrosis, and inflammation, which is seen in both animal and human studies. Paradoxically, however, just like the disease populations, there are healthy individuals, like middle-aged, veteran, intense endurance exercise athletes, that also get high rates of atrial fibrillation. The constant demand for the blood to the working muscle causes the heart to augment its output, leading to increased diastolic pressures and sustained pressure gradients then between the atria and the ventricle near the end of the filling phase. This leads to a stretch stimulus of the atria. But importantly, the left atria and ventricle see different amounts of stress, as the left atria is a much thinner chamber than the thicker left ventricle. Therefore, just like it's easier to blow up a thinner balloon than a thicker one, the atria will stretch and become enlarged compared to the ventricle. And over time, with constant stress, they become inflamed and undergo remodeling, which is seen in animal models. And in fact, previously, our lab showed that 6 weeks of intense swim exercise in mice actually caused these changes and atrial fibrillation, with minimal changes occurring in the ventricle. So populations on the opposite sides of the health spectrum have high risk of developing atrial fibrillation. But what's the common link? Well, the common link is that there's elevated filling pressures, which translate to the disproportionate atrial stretch and inflammation. Recent AF studies in aged hypertensive mice with valvular disease also show that macrophages increase in the atria of these mice. But funny enough, again, and aligning with the classical literature, the valvular disease seems to be the strongest component that increases macrophage numbers the most. So clearly these macrophages can sense and transduce signals in response to stretch. But how? Well, other mouse studies show cardiac macrophages express the mechanosensitive receptor TRPV4. And when activated, this can signal to increase IGF-1 expression that can influence adaptive remodeling downstream. So these cells can sense and transduce the mechanical stretch and are associated with adverse remodeling outcomes. But they also have other important roles in homeostasis and disease, such as facilitating electrical conduction in the heart through gap junction coupling, or phagocytosing debris and apoptotic cells, or regulating inflammation through secretion of pro and anti-inflammatory factors. Now, classically, macrophages were kind of categorized into two broad categories, being M1 inflammatory and M2 anti-inflammatory macrophages. And they were all thought to arise from circulating monocytes, but this has been shown to be a gross oversimplification. Recent lineage tracing, RNA-seq, and flow cytometry data from Dr. Eppelman and Dr. Dick's groups informed us that there's at least 3 distinct subsets of tissue-resident macrophages that have different origins and different functions. For example, there are embryonically derived pro-reparative macrophages, which are entirely self-renewing, which are called TLF positives. There's also another one that is partially replaced by monocytes, and is responsible for interfacing with the adaptive immune system and responsible for antigen presentation. That's called the MHC2 positive TRMs. And finally, there's a subset called the CCR2 positive TRMs, which are highly pro-inflammatory and fully replaced by monocytic input. Um, Dr. Dick's group also showed us that in the heart, you can best parse these groups based on surface expression of TIMD4 and CCR2. Notice here, however, that there's one group that they didn't really highlight that exists in the steady state heart, but we'll get back to that. Also, there's other mouse studies that show that chronic remodeling outcomes in hypertensive mice can be modulated with macrophage interventions. So therefore, this leads to my hypothesis that myocardial stretch caused by increased filling pressures is a potent stimulator of cardiac macrophage and immune cell responses, which can sense and transduce these signals and influence cardiac remodeling outcomes and ultimately atrial fibrillation risk. But there's two things to remember. Despite these links between stretch and immune cell changes, nobody has looked at the acute time points. They all look at the chronic outcomes. And also, most of these groups look at either the whole heart itself or ventricles only without paying attention to the differences in cellular response in the more compliant atria versus the stiffer ventricles. So to test this hypothesis, Dr. Robert Lakin from our lab has developed and characterized a surgical model of valvular disease, which again is one of the highest risk factors of atrial fibrillation called aortic regurgitation. So in aortic regurgitation, we perforate the aortic valve using a catheter that's introduced through the carotid artery of a mouse under echocardiographic guidance. When we perforate this valve, it leads to an immediate backflow of blood from the aorta back into the left ventricle, which increases left ventricular pressures that will be transduced to the atria during the filling phase and cause stretch. To prove this, we used hemodynamic telemetry implants in our mice, and we showed that the filling pressures peak at 3 hours, around 3 hours, but then they progressively decline over time, but remain elevated compared to the sham mice. This acts as our potent stretch stimulus. Then using flow cytometry, I perfused and extracted the heart to clear it of blood and then separate the chambers and digest it into a single cell suspension to tag with antibodies and check for immune cell populations and how they respond. Starting with the atrial data, we see that there's a 10-fold increase in the total macrophage numbers with an 80-fold increase in pro-inflammatory CCR2 positive macrophages and a more modest 10-fold increase in the anti-inflammatory CCR2 negative macrophages compared to the control animals. I further used immunohistochemistry using the macrophage markers F480 and CCR2 to check how these subsets change. And they agree with my flow data as we see an increase in the total F480 cells and the inflammatory CCR2 positive cells with our 1 day of stretch stimulus. Next, following the more recent literature, I used TIMD4 and CCR2 to parse my macrophage populations into the respective groups. Surprisingly, I found that 1 day of AR was enough to cause increases in all the macrophage subtypes throughout the line. So 1 day of stretch stimulus is enough to cause a profound macrophage response, but it also causes a macrophage polarization into a greater proportion of CCR2 positive cells, which are more pro-inflammatory in nature, as shown by these pie graphs. Unexpectedly, however, now coming back to this graph, we also saw a large 70-fold expansion in the understudied group, which they didn't really mention or they didn't really expand upon in steady state conditions, and it hasn't been explored in disease conditions. Interestingly, the immune response in the atria were not limited to macrophages alone. We found a massive infiltration of Ly6E high classical monocytes, which makes sense if they're going to be replenishing the CCR2 positive and MHC2 positive macrophages which are known to be replenished by monocytes. We also saw an increase in neutrophil levels in the atria, but using further histological analyses with hematoxylin and eosin showed that there was no visible evidence of tissue damage, as evidenced by no presence of pycnotic nuclei or myocardial fiber disorganization. Therefore, a pure stretch stimulus to elevated filling pressures was enough to bring all of these cells into the tissue. To try and determine where they came from and localize, we further analyzed the localization of these cells using IHC and a stitch of the atria. And we found that the F480 positive macrophages and CCR2 positive cells primarily localized in the subcapsular region of the atria. And this could indicate that this is where the stress and stretch of the atria is felt the most. In contrast, although the left ventricle also saw an increase in total macrophages, it was about twofold less compared to the more compliant atria. And there was a 50-fold increase in CCR2 positive macrophages and 4-fold increase in the CCR2 negative macrophages, which is still more modest compared to the 80 and 10-fold increase in the atria. Furthermore, a greater difference in response between the chambers is seen when you parse these macrophage populations by their TIMD4 positive TIMD4 and CCR2 surface marker expression. So although CCR2 and MHC2 TRM saw somewhat of an increase similar to the atria, there is still a non-significant and less than twofold expansion of these embryonically derived TLF positive macrophages, which in comparison to the atria expanded almost fourfold and significantly. Additionally, there was only a 15-fold increase seen in this understudied TIMD4 positive CCR2 positive macrophage group compared to the atria, which saw a 70-fold increase. So clearly, the difference in chamber property is what's causing or may be causing the differences in the cellular response to the stretch. I further used the IHC in the ventricle as well, just to see if it aligned with our flow data. And we saw a non-significant increase in the total amount of F480 cells and slightly less CCR2 positive cells increase compared to the atria. Further analysis showed that the monocytes and neutrophils, which also infiltrated, were to a lesser extent than the atria. Finally, since infiltrating monocytes are thought to entirely replenish the CCR2 positive macrophages and partially replenish the MHC2 positive macrophages, we looked at the Ly6C status of our macrophage groups. So when we did this, we found that, as expected, monocytes were significantly replenishing both the CCR2 positive and the MHC2 positive macrophages in the heart in both chambers. And also expectedly, the self-renewing subset did not see any significant increase in the Ly6C status, meaning that they weren't really expanded with monocytic input. However, unexpectedly, we found that the group that's understudied in the steady state and disease conditions actually did see an increase with monocytic input. So to confirm this finding, we wanted to make sure that the monocytes were actually replenishing these macrophage groups. So we ended up using a CCR2 inhibitor. Now, the CCR2 inhibitor or CCR2 itself is a chemokine receptor that's required for monocyte egress out of the bone marrow and subsequently tissue infiltration. So when we use this inhibitor, we found that total amounts of macrophages alongside all macrophage groups were attenuated with one-day AR. So that means that whatever monocyte input was coming in to replenish these macrophage groups were CCR2 dependent. To conclude, myocardial stretch especially from valvular disease, is a potent promoter of cardiac inflammation. One day of aortic regurgitation induced a pure mechanical stretch stimulus, which again is likely not attributed to tissue damage, causing a profound increase in total left atrial total macrophages, all of their macrophage subsets, and classical infiltrating monocytes. And finally, the left ventricle sees a relatively less pronounced immune cell response in comparison to the atria, most likely due to differences in compliance between the chambers. Macrophage expansion to acute AR and stretch was monocyte dependent and was attenuated with the use of the CCR2 inhibitor. Yeah, and that is the end of my talk. Thank you so much for listening and your time.
Thank you so much for that. That was an amazing talk, really informative. Um, I just wanted to let everybody know if they want to ask Parashar a question, we have 5 minutes for a question and answer period right now. Just raise your hand and unmute yourself, and you can go ahead and ask your question to Parashar. If nobody has a question, I have one.
Sure.
Um, I was wondering, you mentioned these other TRMs. Do they have a label or like what is encompassed in that umbrella term? I want to ask.
Yeah, I mean, so let me go back to it. Right. So in this graph, right, what they did is first they selected for in flow cytometry, what you do is you select for all of the myeloid immune cell subsets using CD11B or a surface marker that's attributed to just that subset. Then you'll parse it further, saying that, okay, if it doesn't express Ly6G, for example, it's a non-neutrophil. If it expresses CD64 and F480, for example, it's considered a macrophage, right? So using this way of narrowing down on our cell population, um, and the fact that it expresses both TIMD4 and CCR2, we can find the cell populations in our heart. But to answer your question, um, the fact that it expresses TIMD4 means suggests that it's an embryonically derived subset. But the fact that it expresses CCR2 means that it must be recruited or it's coming in somehow. Therefore, I think that this subtype is actually coming from a different source. And that's what I'm studying now. Like I'm working on these studies. But nothing is really known about the function of this group
Mhm.
in both steady state or disease, right? So it's a very interesting finding.
That's really cool. Thank you so much.
Yeah. And I mean, like it's very hard to find evidence on this group. This graph particularly, we can see that they just, for whatever reason, they just chose to ignore. They ignored 5%. So if you add up these numbers, it equals 95%. They just didn't like delve into this 5%. But this is the group that we found expands the most, which is a little bit unexpected.
Yeah, very interesting. That's awesome.
Great. I have a question.
Yes. Yes.
Yeah, great. Yeah. So very, very interesting presentation. So you studied this at one day with the aortic regurg model, yeah? So
Correct.
were these mice in atrial fibrillation at that stage, or was this a pre-AFib state?
Yeah, this is a pre-AFib state. We have, like Dr. Robert Laking in our group has
Uh-huh.
actually characterized this model fully. So he's gone to the chronic time points and shown that these hearts get hypertrophic and they get atrial, they have 60% inducibility for atrial fibrillation, right? So right now, the point that I'm trying to make is that the changes which occur in the atria may be facilitated by these immune cells which are responding to the stretch early on. They might dictate how the remodeling is sort of continued on into the later stages.
Mm-hmm. Yeah, very good. How do you propose the macrophages are creating a susceptibility substrate for AFib.
Right. So macrophages are known to um, they sort of modify the environment when they secrete their pro-inflammatory factors or pro-reparative factors, and this makes this allows other cells to respond and maybe deposit fibrosis to a greater degree in one chamber than the other. But when that happens with the more chronic time points, the fibrosis acts as a conduction block, and the hypertrophy makes the wavelength greater in the path length greater in the atria, which are all key hallmarks of atrial fibrillation. So they may mediate the maladaptive remodeling later on.
Okay, great. Thank you very much.
Thank you. Sabrina, I see you have a question.
I do. Hi. Thank
Hello.
you so much for such a great talk. Honestly, as a novice, this is all very interesting to me, and I have quite a few questions, but
No
I
problem.
think the most applicable question here I have is if we were to take this in a model of, let's say, skeletal muscle, after exercise, you said the three major subsets of resident immune cells we have are the TLF positive, MHC2 positive, and the CCR2 positive. If
Correct.
I were to look within skeletal muscle after an acute bout of exercise, would I also find these resident immune cells to facilitate some of those either positive or maladaptive adaptations you were talking about, or is this really
That's a
specific
great
to?
question, and actually that speaks to how translatable is this, right? So I actually prepared this beforehand. Um, can you see my screen?
Yes.
So this group, Sarah Dick's group, actually checked other organs and like skin tissue and even muscle. So these groups are actually conserved within almost every organ, right? So they
Mhm.
exist within skeletal muscle, like in the hind limb, in the diaphragm, the tongue, etc. And they've done RNA-seq data to show that they have a similar function in these different places as well. So it's
Okay, great.
very possible that this could expand with acute bouts of exercise in skeletal muscle.
Do
Absolutely.
you have any plans to examine that in your hands and confirm that, or is that not something you guys are interested in?
Not at the moment. Where I'm trying to kind of figure out where this fourth macrophage group kind of comes from and what function it
Hmm.
plays first. And you can even see that within the different organs and skin plate, like within the different locations, they actually do have the evidence of this fourth group. But again, so they just chose to not discuss it.
I mean, I think the concern here, I guess, for those people that don't do flow is how are these gating strategies really achieved, and how can you know that that certain population that you're looking at is the specific population you're talking about? So, do you think you could just maybe walk us through a little bit about from tissue collection to immune cell
Yeah,
isolation
sure.
to your gating strategies?
Absolutely. So we start by perfusing the hearts very well. So I take a syringe and I have a couple buffers. I have an EDTA buffer and a perfusion buffer. It doesn't really matter what it is, but the fact is, when I get it out of the mouse, right, when I take the heart out of the mouse and put it in the dish, I further perfuse it to get rid of any blood, right? Because blood contaminates our idea of what's in the tissue. So if there's
Mhm.
any blood remaining left in there, it might dilute what we're seeing. Once that blood is gone, I will separate the chambers into the respective chambers, the left atria and the left ventricle, and then digest it with enzymes and I'll mince them into little pieces so that it helps with the digestion into a single cell suspension. Once I run it on the cytometer, or sorry, right after I make it into a suspension, I have to tag it with antibodies. Now, these antibodies are specific to certain cell surface markers, right? And these cell surface markers are only attributed, like some of them are only attributed to certain cell types. So, for example, CD64 plus F480 makes it only a macrophage, right? Or Ly6E high means that it's monocytic or monocytic in origin,
Thank you.
right? So, through this method, we can now take it to the flow cytometer. Once we run the sample, we can electronically gate out those cells which are high in, I don't know, like width and height, which ends up being aggregate and debris. So you can get rid of that and just focus on what immune cells there are. And then after we gate on CD11B, which is our pan-myeloid marker, then we can further refine and say, okay, which ones are neutrophils Ly6G positive and non-neutrophils Ly6G negative, and which ones of these Ly6G negatives are now macrophages CD64 positive or monocytes CD64 negative, and then so on. But this is a much more sensitive way to kind of narrow down on your cell of interest compared to IHC. Because in IHC, you only have maybe maximum 3 markers that you can look at.
Very cool.
Just
Thank you.
Thanks so much, Parashar. We do just have to move on to our next speaker, just so everybody is aware. If they weren't able to ask the questions that they want to ask to Parashar or Amanda, our next speaker, there will be breakout rooms right after the next question and answer period, so you can join those speakers' breakout rooms and ask further questions. Thank you so much, Parashar, for your talk.
Thank you, everyone. Thank you.
I would like to introduce our next speaker, Amanda from the University of Alberta. Amanda, if you could just share your screen and take it away.
Can you guys see my screen? Okay, thank you so much, Fesha. That was a really interesting talk that you gave, and I'll be sure to join your breakout room. Okay, so to get into my project, I'll be focusing on a HFpEF mouse model. So before I get into the meat of my project, I'll just give a quick overview. So first, in order to understand my main topic, heart failure is defined as a condition in which the heart muscle cannot pump enough blood to meet the body's energy and oxygen requirements. And this can either occur acutely, suddenly, or can develop over time, be a chronic condition. And heart failure can be caused by many different things, from coronary heart disease, hypertension, infections, or diabetes. And it is largely divided into 2 different kinds: diastolic, also known as HFrEF, or systolic, which is also known as HFpEF. HFrEF is heart failure with a reduced ejection fraction, whilst HFpEF is heart failure with a preserved ejection fraction. And ejection fraction is defined as the amount of blood left that the left ventricle pumps out in a beat. And as you can see in my diagram on the right at the top is a healthy heart, and the way that the blood is pumped in a healthy heart is first the blood enters from pulmonary veins into the left atrium, into the left ventricle, out through the aorta to go out to the rest of the body and provide oxygen to the rest of the body. And when the deoxygenated blood returns to the heart, it enters in through the vena cava, in through the right atrium, in through the right ventricle, and back to the lungs to be reoxygenated. So then in the case of a HFpEF heart, when the blood enters into the left ventricle, it's much harder for it to do so because the heart muscle is stiff and thickened. And in the case of a HFrEF heart, the blood can enter the left ventricle correctly, but then it's much harder for the blood to be pumped out because of a dilated left ventricle due to a weak heart muscle. And then moving on to sarcopenia, as it's also pertinent towards my project. Sarcopenia is defined as a progressive loss of muscle mass, strength, and function due to age. And sarcopenia is highly prevalent in Asian populations, as is heart failure. So these two kind of combine to exacerbate each different condition. Sarcopenia is caused by the lack of exercise, poor nutrition, chronic diseases such as diabetes, and again, heart failure. And as you can see on the bottom left is just kind of a quick schematic of what sarcopenia is. As you can see on the left is a healthy, young muscle tissue where there's a lower percentage of fat and a higher percentage of muscle fibers, whilst to the right is the fat then replaces the muscle fibers, there's more fat and less muscle, which leads to weakened strength and function of the muscle tissue. Okay, next I shall give a quick overview of a very important peptide in my project, which is called apelin. Apelin is an endogenous hormone that binds to the G protein couple, apelin receptor, also known as or APJ. This signals, this peptide signals for cardioprotection and helps regulate vascular tone, angiogenesis, and gene metabolism. This peptide often acts like a counter-regulator to the RAS system, which is often overactivated in conditions such as heart failure, hypertension, disease. Apelin has been found to help promote ACE2 production. It cleaves angiotensin II, which then promotes vasodilation, which helps in hypertensive conditions. And apelin has also been found to apelin deficiency has been associated with reduced myocardial contractility, exercise capacity, as well as decreased myocardial angiogenesis and impaired skeletal muscle mass and metabolic function. A paper by Vinil et al. Nature Medicine has also confirmed these results. The importance of apelin as an important exocrine. But the challenge that arises is that native apelin peptide shown at the top right of the screen is that this peptide has a short half-life and is very quickly often degraded by multiple different proteins such as neprilysin, human basic kallikrein, and ACE2. But to combat this, our lab has developed a protease-resistant apelin analog for testing our project, shown at the bottom right of the screen. And next, I'll just go over some of the therapeutic potential of apelin. And as mentioned earlier, apelin binding enhances cardiac contractility and reduces myocardial damage. It also helps with renal dysfunction, hypertension, and promotes angiogenesis. And apelin levels have been reported to decrease with age. And again, apelin deficiency results in reduced skeletal muscle mass and function in mice. And the supplementation is often associated with improved muscle regeneration, contractility during exercise, and enhanced mitochondrial function and biogenesis. And this is especially relevant in the context of GLP-1 receptor agonists, which is often referred to obese patients as weight loss medicine, as well as for HFpEF patients. But then the problem that arises is that some studies have shown that GLP-1 receptor medicines may lead to muscle mass loss. Then onto the main project design of the study is that the main focus of the study is to investigate the impact of the loss of cardiovascular system as well as skeletal muscle in aged diabetic mice. We are using 7 to 7.5 month old black 6 mice to simulate muscle wasting and to replicate a HFpEF phenotype. And we use both male and female mice to account for any sex-based differences. And the 4 different experimental groups are wild-type mice as a control, a global apelin knockout just to observe the effects of apelin alone, DB diabetic mice to see the effects of diabetes without compounded without the loss of apelin, and DB apelin mice to see combined effects of both diabetic phenotype and the loss of apelin in both the cardiovascular and muscle systems. And to the right, you see a diagram of a quick schematic of the timeline that is followed through the study. The first of the mice are bred around 4 weeks is genotyping just to confirm the genotypes. Then at 12 weeks is when we do the monthly weigh-in to division until the final endpoint of 32 weeks, where we do our testing such as metabolic testing, cardiac, vascular, skeletal muscle testing. That's when we also do our collection. Okay, onto some of the data that we've got so far. As you can see on the left graph, this is an image of the injection fraction between 4 different groups. And see the injection fraction of the wild type, Evelyn, DB and DB8 groups are fairly consistent between the groups, so no significant difference between them. And this just confirms the HFpEF phenotype because the issue is the actual filling. Like the issue isn't with the ejection fraction, but with the filling of the heart. And on the right, the top right, is an M mode image of the left ventricle. This measurement, if you, not sure if I can zoom in, but the LVPW, which is the annotated LVPW, is the left ventricular posterior wall during diastole, which just shows the muscle thickness of the left ventricle. As you can see that there is not that much of a significant increase in the DB8 group, but there's a slight higher thickness in the DB8 group, which again just confirms that this is a
Sorry, Amanda, just 1 second. I noticed that your mic is cutting in and out a little bit. Maybe switch to your either like if you have headphones in, switch to your computer mic just to make it a little bit
Okay.
easier for her to understand the data slide. Sorry.
Okay. Can you hear me now? Okay.
Yes, perfect. Thank you.
Yeah, sorry about that. Um, okay, moving on to the diastolic function. In the middle row, the top image that you see is a pulse wave Doppler image of the filling velocity of the left ventricle, which is annotated as the capital letter E. And with the second image in the middle, what you can see is a tissue wave Doppler image with the filling pressures annotated as E prime. And with both of this E over E prime ratio, this shows describes the left ventricular filling pressures with a HFpEF model. These ratios are often increased, and as you can see with our graph on the left, is that the DB apelin group is approaching significance, slightly compared to the wild type apelin and DB groups, and potentially with more data points, we'll be able to like really tighten it and hone in on the significance. And another marker of HFpEF is an enlarged left atrial dimension. And with this M mode image of the left atrium at the bottom, you can see, right? At the bottom, M mode is an M mode image of the left atrial dimension. And as you can see, once again, the DB apelin group is shown to have a slightly greater degree of left atrial diameter, hinting towards a HFpEF phenotype. And moving on to some of the muscle testing that we've done so far, the image at the top left, we performed an echo MRI to assess the body composition of the different mice. And we see with the DB group, it has a higher lean mass than all the other groups. And for the remaining 3 graphs that you see at the top right and the 2 bottom images, what we did for these graphs is that we separated out 3 different muscle groups, the soleus, the gastrocnemius, and the quadriceps. And firstly, at the top right, you see the soleus muscle group. The DB soleus muscle is significantly lower than both the wild type apelin and the DB apelin groups. And this potentially is due to the low sample size because we only have two DB muscles as of right now, but hopefully with more muscle collections, we'll be able to see more of a significant difference between the groups. And with the quadriceps muscle at the bottom left, what we can see is that between the wild type and apelin groups and the DB apelin groups, there is no significant difference. However, you see when you compare the DB and DB apelin groups, they're much lower than the wild type apelin groups. And this may be due to the effect of diabetic muscle loss rather than the effect of the loss of apelin. Otherwise, the apelin group would be much lower than the wild type group as well. And for the results of the quadriceps muscle mass seen at the bottom right, it is similar to that of the gastrocnemius group. What we can see is that the DB and the DB-apelin groups are significantly lower than the wild type and apelin groups, but we're not seeing so much of a muscle loss due to the lack of apelin. So we'd have to conduct some more tests and do some histology staining just to really hone in on what it is exactly that is causing this discrepancy. And onto some of our metabolic results. So at the left, you see the results from our oral glucose testing, and this shows that the glucose handling is not impaired by the loss of apelin, as the wild type and apelin groups have similar glucose tolerance curves, as well as the DB and DB-apelin groups. So this just shows that the glucose handling is impaired by the fact that the mice are obese and not because of any loss of apelin. And this result, once again, is confirmed with another body composition image shown to the right, where the wild type and apelin groups have similar fat mass, whilst the DB and DB-apelin groups have similar to each other, but much greater than the wild type and apelin groups, which makes sense because these mice are obese, whilst the wild type and apelin groups are not. And in conjunction to the loss of function study, we also decided to do some gain of function studies just to assess, just to really hone in on what it is that's showing the discrepancies. And I'll just do a quick overview of the project design again. So what we did was use 26-week female black 6 mice. We placed them on a high-fat diet and aged them out to 65 weeks. And once they've reached the 65-week mark, we begin to supplement these mice with either apelin or a saline placebo, and then do testing, a normal testing like echo MRI, echocardiogram, PV loop, tissue collection and histology at 17 weeks, just to confirm whether, just to assess whether it has a HFpEF phenotype or not, and just to see whether apelin supplementation does help rescue this phenotype. And onto some of the results. Once again, as with the loss of function study, you see in the graph on the left, the ejection fraction between the three groups, the control, saline, and apelin groups, that this is fairly consistent. There's no significant changes between the groups, and this just confirms our HFpEF phenotype. And with the measurement on the right, this is the left ventricular internal dimension during diastole, and which is just this is just a measure to assess how dilated or how large or small the left ventricle is. And as you can see, again, there's no significant difference between the groups, which means that the left ventricle is not dilated, which just confirms that this is not a HFrEF phenotype, but a HFpEF phenotype. And onto some of the diastolic function. Again, on the left with the left atrial dimension, both the saline and apelin groups are greater than the control, whilst the saline group is approaching significance with comparison to the apelin treatment group. We may have to do some more studies on this, but it's looking slightly positive. And then looking at the right, in the mitral valve E over E prime ratio, this just shows that our apelin treatment group is lower than that of the control and the saline treatment groups, and this suggests that the apelin supplementation helps rescue this phenotype by reducing some of the hypertrophy. And next, I'll just do a quick overview of where we are now and how we may move forward. And with the loss of function study, we've confirmed that the model successfully replicates a HFpEF phenotype. We see no significant systolic function changes, and we see that the loss of function groups have greater E prime ratio, which suggests greater filling pressures. And so far, there have been no significant markers of sarcopenic muscle mass loss, and this may be due to insufficiently aging out the mice. And for the gain of function study, once again, the model successfully replicates a HFpEF phenotype, and we see a decreased mitral valve E over E prime ratio, which suggests better diastolic function with apnea treatment. And we also see a decrease in left ventricular posterior wall thickness in 70 weeks with apnea treatment, which again suggests reverse remodeling of the hypertrophic phenotype. And I'd like to acknowledge my lab mates and the sponsors that we've had. And thank you for listening to my presentation.
Great presentation. Thank you so much, Amanda. We do have 5 minutes for any questions for Amanda and that amazing presentation. Thank you. Again, you can just raise your hand if you have any questions and unmute yourself and go ahead and ask the speaker.
Hi, Amanda. Really great job. I really enjoyed your presentation, so thank you so much. I was just wondering, obviously you have a lot of really great preliminary data, but do you see apelin supplementation having any other application other than, sarcopenia and the perturbations to the cardiovascular system that you were discussing? Is there any literature to support its role in a different setting? Just wondering to learn a little bit more about it.
I mean, our lab focuses mostly on the cardiovascular and skeletal muscle, but some literature does suggest that apelin does have a role in kidney function. So we've looked at maybe doing some renal function testing just to see if there is a difference between the two groups of like apelin supplementation versus not supplemented by apelin.
Yeah, it makes you think a little bit about the organ crosstalk when you're supplementing any agent to a whole body model. So I think definitely some merit to that, and maybe other groups that may be interested in collecting tissue from you in a different setting. That could always be really interesting. So nice to hear that. Thank you.
I do have a question. I was just wondering, did you use the same age between your gain of function and loss of function studies?
No, we did not. Although, especially with the sarcopenic phenotype, I presume that our data isn't as striking because they're not as old as they could be. But then the issue is that as we begin to age out our mice even more, is that they start to become like develop lots of microencephalies and eye diseases. So they're not very viable past a certain point. So this is about the furthest we can push them, unfortunately.
Yeah, I found it really interesting with aging research. It's like the models are very different each time. So it was really interesting to see that you decided to use two different models. Um, Really cool data. I also have another question if nobody else has another question. Um, I was wondering, I might have just missed this. How did you collect your full body composition data?
Oh, we use an echo MRI machine. So we just place our mice in a tube and put the tube in the giant machine, and then the machine uses the magnetic resonance imaging just to give us the data back on what percentage of the mass is lean tissue, water, or fat mass.
Oh, very cool. Okay. Thank you.
Mhm. No problem.
All right, so we are going to just go into our breakout room. So if you have any questions for either of the speakers, please join their breakout room. I am going to open all the rooms. You can ask the participants or you can ask the speakers any questions that you would like, and you can just put yourself in either of the rooms.